of microbiology, that is immunity. There are two types of immunity. First one is a cell-mediated immunity and second one is a humoral immunity. So cell-mediated immunity is used by T lymphocytes or T cells, and humoral is used by B lymphocytes or B cells. So first of all we will take antigen. This antigen will presented to B cells, and then the plasma cells will activate and antibodies are formed. This whole process is a humoral immunity, as it is controlled by B cells. Then other antigen which is presented to antigen presenting cells, then T helper cells are activated, then it releases cytokines and inflammation takes place, and this inflammation and will react with the antigen. So this is a cell mediated immunity as T helper cells are involved. First step is induction of cells, that is B cells or T cells, and the last step is reaction. That means the product formed here, antibody, and here inflammation, will react with the antigen. Then types of antigen. There are two types of antigen. First one is a complete antigen and second one is an incomplete antigen. So complete antigens, they can induce antibody formation and can react with the antibody. Means these antigen can form antibody as well as they react with antigen. This antibody form can react with antigen. Means the complete antigen will react with the formed antibody. Incomplete antigens, they are also known as heptans, that is, they do not induce antibody formation as their size, as here you can see this is a complete antigen, its size is so large, and this is incomplete antigen. So our body cannot process this or recognize this antigen but can react with the antibody. Here its size is larger, so it can induce as well as react. Molecular weight is less than 100,000 kilodaltons. They are not immunogenic, but they become immunogenic when linked to the larger carrier protein. There is the heptans, which are the smaller or incomplete antigen. They combine with the carrier protein and form complete antigen. It means we can convert incomplete antigen to complete antigens. Then adjuvants. Adjuvants are nothing but the substance which are different from antigen, but when mixed with the antigen, they increases the immunogenicity of antigen. For example, as we know, alloy, which increases the antigenicity and it can be used in certain vaccines. Then the pertussis vaccine, as we know, DPT vaccine, diphtheria, pertussis and tetanus. Here pertussis increases the immunogenicity or effect of diphtheria and tetanus. Then we will deal with the epitope and paratope. Epitope, here we can see the antigen and it is the antibody, which is a lambda chain. It has heavy chain, light chain. So epitope is a part of antigen which react with antibody, and paratope is a part of antibody which react with antigen. So epitope is also known as antigenic determinant, as it is a part of antigen. Immunologically active regions of an antigen is called as epitope, that actually binds to the specific receptor of antibody. It is the smallest unit of antigenicity. Then the paratope, it is the combining area of the antibody corresponding to the epitope. Specific antibody determines to the paratope and called as ioidotopes. Epitope and paratope determine specificity of immunological reactions. These epitope and paratope are specific in nature, as this epitope will only bind to its subsequent paratope and bind to the certain antibody, not to the referent antibody, as we know, a lock and key model. Then the types of epitope. There are two types of epitope. First one is a linear or sequential epitope. and second one is the conformational epitope. Linear epitope, as you know, it is a linear chain formation. It's formed by the amino acid which are adjacent amino acid. Then conformational epitopes are formed by the amino acid but not in sequence, means its structure is certainly different. So linear subsequential epitope will identified by T cells and the conformational is identified by the B cells. Then determinants of antigenicity, it is a very much important topic. So determinants of antigenicity, there are 10 determinants of antigenicity. First one is the foreignness, molecular size, chemical nature, physical form, degradability, route of administration, genetic factors, specificity, autospecificity and heterogeneity specificity. First one is the foreignness. जितना ज़्यादा फॉरेन होगा एंटीजन उतनी ज़्यादा होगी उसकी एंटीजेनेसिटी. और सब्सटेंस आर फॉरेननेस टू इम्यून सिस्टम, वो हमें कैसे पता चलेगा कि दिस सब्सटेंस इज फॉरेन टू आवर इम्यून सिस्टम. सो अकॉर्डिंग टू द बर्नेट्स क्लोन सिलेक्शन थियरी, फॉरेननेस दैट इज नॉन सेल्फ means substance that never come in contact with the lymphocytes during the embryo period. So as we know, this is the embryo is a blood vessel, so lymphocytes are present inside the blood vessels. The substances which come in contact with these lymphocytes, they are considered to be self, and the substances which do not came in contact with that lymphocytes, they are consist to be foreign substances. Then second one is the molecular size. More the molecular size, more will be the antigenicity. Minimum 10,000 Dalton से बड़ा साइज होना चाहिए. Then less than 5,000 Dalton will be non-antigenic, considered to be non-antigenic. Then third one, nature, that is protein will be more antigenic as compared to carbohydrate, lipids, and last one is the nucleic acid. Then physical properties, as we take a solid and liquid, solid will have more antigenicity as compared to liquid. Then degradation, more the substance will degrade, more will be the antigenicity, as its epitope, paratope will be more visible as it get degenerated. Routes of administration, as we take antigen A, so it is given IV, subcutaneous and oral. Subcutaneous will have more antigenicity as subcutaneous will in contact with the lymph nodes, so it will have more antigenicity. Then the genetic factor, some substances can be immunogenic to one person and cannot be immunogenic to other, it depends on genetic factor. Then autospecificity, the substance which came in contact with the blood, as we know by the law that the substance which came in contact with the blood are considered to be self, and the substance which are not in contact with the blood, they are non-self, that is they are foreign. For example, the eye lens which is that is cornea of eye, which do not contain blood supply, which do not have blood supply, so it will be foreign to the body. Then heterogeneity or heterophilic antigen means different species. As we continue this bacteria will have certain proteins present, so antibodies form against this, but it mimic on present, means similar proteins are present on the human cell, so it will degrade this different biological species. एंटीबॉडी मिमिक करेंगे, अपने ऑर्गन को भी डैमेज करेंगे. देन लास्टली वी विल डील विद सुपर एंटीजन. सो दिस इज नॉर्मल एंटीजन, it binds to the antigen presenting cell. Then T helper cells, cytokines are released, then it will activate the B cells and antibodies are formed. This is normal antigen. Super antigens, they do not attach to the antigen presenting cell, they directly bind to the T helper cells, and they increases number of T helper cells that is T cell receptor. They do not follow the T cell receptor rule. They activate all the T cells, means maximum T cell, and massive cytokine is released. B cells are activated in more numbers and antibodies are formed in more numbers. So hypergammaglobulinemia can occur. Then the activator of T lymphocyte without relation to their epitope specificity is not followed. They activate the T cells without being processed by the antigen presenting cell, massive release of cytokines and increased antibody production, that is hypergammaglobulinemia. So here are the 10 types of super antigens. First, second, third, 4 are bacteria, 4 are viruses and 1 is a fungus. You have to learn all this. First one is streptococcal toxin, toxic shock syndrome 1. Then mycoplasma arthritis, Yersinia enterocolitis, Epstein-Barr virus associated superantigen, cytomegalovirus, then rabies virus, HIV encoded, malassezis, purpur.